All right. Okay, then let's start today's seminar session. It's my pleasure to introduce Hélder Santos from the University Medical Center Groningen, who's visiting us today in the frame of a project, a European project that we're working in, and also in the frame of the DocFam seminar series. Just a little bit about the background of Hélder. He obtained his master's in chemistry from the University of Porto, and then moved to Finland to develop his career, where he obtained his PhD in chemical engineering from the what is Aalto University today. And then after some postdoctoral work, he was appointed associate professor in 2019, and then full professor in pharmaceutical nanotechnology in 2020. And after which he moved then to Groningen, where he is now full professor and chair in biomedical engineering, and also head of the Department of Biomaterials and Biomedical Technology. So Hélder's research interest, as you can also see from the title of today's talk, is the development of nanomedicines and biomaterials for biomedical applications, and particularly focused on cancer and heart diseases. He's also co-founder of two startups and has published more than 550 publications, and also holds 6 patents. So it's my pleasure to have you here today, and we're looking forward to your talk. Thank you, Hélder.
Thank you very much, Mariana, for the kind introduction. It's working, I hope. So thank you very much. It's a pleasure to be here today with you. I've never been here, so this is my first time, and I'm enjoying really a lot what I have seen so far. And also thank you so much for your hospitality. So today I put here a talk also to show you what we are able to do and how we could probably find ways how to collaborate. But let me, of course, start. This works? No? See? Yeah. So let me just start to tell you where I'm coming from. So this is Netherlands, a small country. That is Groningen, so we're about 2 hours from Amsterdam. And actually, my department is part of the University Medical Hospital. So the university has, the medical hospital has about 13,000 people working there. Most of them are doctors and nurses. And there is also plenty of researchers, but basically there is a good setting to interact between the preclinical and the clinical side, because you find a lot of medical problems, and that's how we try to solve. So it's before I came there, it was more on what we can do or what we can do for science. Now it's more what we can do for our colleagues, how they can use our materials, what are their problems, and how we can help our clinicians to solve the medical problems. So the department is called Biomaterials and Biomedical Technology, and I will show you what we are able to do. But basically, this is the hospital. So here on this part, we have all the clinical parts, so with patients. And then on this part here, we have all the preclinical side. So this is where my department is located. So we have 2.5 floors here on this building, on the 13th floor. And everyone that visits my office says that I have the best office in town, because I can see the clinicians down, and I can see the whole city around. So that's a perfect place. But that is a nice setting, because you can just cross this bridge here, this building here, and you are discussing with your clinicians, and you can interact quite a lot with them. So the department itself, so this is my department. We are about plus 100 people, and we have different groups there, and we go from physics to chemistry to robotics. So we have different kind of settings there, and I will show you later. Most of the department are about 60 people are PhDs, or 65. Then we have few postdocs, technicians, and then about 11 PIs. So what we do, so this is shown here. So we go from the material interactions, techniques to learn how these materials interact with tissues and cells and interactions, drug delivery systems, imaging and analysis systems as well. So we work a lot with Raman, for example, biomaterials on different way, and also robotics. So we work with small robotics and surgical robotics. So we have a group very heavy on that. And then we work with, well, you will see, but with different kind of nano and microsystems. But basically, our department gives a lot of support to the medical problems. And so therefore, we work a lot with the clinicians. So just some numbers, as I mentioned, we are about 105. Very, very balanced in terms of gender. And we also just last year initiated a company. So this is QT Sense, is based on Raman microscopy, MRI. So a group, one of my PIs, Professor Romana here. So she is commercializing one device so that you can analyze directly from cells or from tissue samples reactive oxygen species. So that's if you are interested, you are very welcome to visit the web page. So just to give you another numbers of our, as I said, we are actually not a very big department when we compare to other departments at the medical school. Yet we have had, so last year, more than 100 papers, for example, 12 PhD theses, and our funding is running mostly on external funding, basically EU grants, but also some of regional fundings. So Groningen gets a lot of regional funding. So recently, we just got from a foundation in the northern part of Netherlands, 18.5 million euros. So that brings close together engineers, clinicians, fundamental researchers, and so on, so that we can bring some technologies to the next level. So our group, now more into what our group does. So we are very interested in different kind of nano tools, basically more for delivery and for disease. But basically we try to work with small drug molecules or biological molecules, as you see here, one of the systems here, in order to solve some of their problems. So stability, bioavailability, targeting. So that's what we are trying to engage on, and you will see a lot of this in my next presentations. Obviously, we are always very happy when nano comes to the spotlight. So as I used to say to my student, nano rocks. Basically, you probably remember of the quantum dots Nobel Prizes, the RNA-based therapies Nobel Prize, and very recent again, the microRNA. So these are things that, or these are news that means that we are doing something that is very current, very up to date. And so we try to use some of the approach that you see here. So we have been working in the past a lot with inorganic materials. So we were a group that was built on porous materials. So porous silicon and porous silica. I think I have seen some of your posters. We have also worked at that. So that's where my group started. We did everything possible with porous materials. We still do. But now we are more going into polymeric materials, specific polymeric particles, and lipid-based materials. But the good thing of being in a group that is very diverse is that allows us to study different kind of properties of the materials, different kind of coatings. So we have tried different kind of coatings. And then we have also analyzed this in terms of their response intracellularly, but also in vivo. And I will show you some examples. So you probably are aware of this. These are some of the most of the systems that got into the clinic, into patients. And as if you can see here, they are variations of very similar kind of materials. Lipid-based materials or LNPs-based materials or other. Our group tries to also test novel materials. So we don't know nothing about them. Do they have potential to go into the clinic or not? And then we try to understand at the cellular level, but also at the in vivo, if they would have some potential. So you will see novel materials. However, we already know how difficult it is to get things to the market, obviously. So last year, together with a postdoc here, Paul Joyce, we put together this beautiful, I think it's a very beautiful paper. It's a kind of a guide paper for researchers, but also for industry. And here you see a lot of people that are worldwide experts in nanomaterials, nanoparticles, from industry and academia. So you probably are familiar with Robert Langer, and you probably know also Maria Alonso. So, and we put together this paper that basically what we discuss is that how can we make this much more efficient from a different perspective. So we call it deliver, because it really follows all the definition, target, essentials of characterization, lead compound, intellectual property, validation, economical and scaling up, and regulatory. So we call this delivery paper. And we discuss a lot on this, so I invite you, if you are interested, to read it there. It's a little bit extensive paper, but I think it gives very good considerations how to move forward even faster the field, especially when you have a new material and that you want to bring it to the market. So this is what we try to do. We don't focus in one material. We try to find properties of a material and then see if that material can move forward. So as you can see here, there are different materials that we have used from the past, light-sensitive materials, or other kind of materials that we have produced, and basically different applications. So you see here cancer, for example. You see here heart, for example. Today I will discuss about heart as well. You see here again cancer. You see here bacteria. So we try to design materials or use materials that we can find an application for them. Obviously, most of the times we need to make a target material like this. So this is what we try to do also. Try to understand how the cells and the particles interact and try to find the best possible ligands or the best possible connections between a particle and between a material and a biological cell or tissue. So this is what we do. And obviously what we want them is to make them targetable. So, and you can do this with, again, as I mentioned, with inorganic materials, with organic, but we also use a lot of what we call hybrid. So we mix both worlds, or then more recently, what we have been working quite a lot is on biohybrid materials. So what we do is that we extract cell components and then we construct this material. So for example, imagine a magnetic particle, and we surround this magnetic particle with cell membrane components, for example. And this gives us a different advantage and properties that we cannot find with the materials. So just to give you a very some of the projects that we work on, I don't have time to tell you into much detail, but just to get an idea what we do, I show you some of the works that we have done in the past. So this is one of example where we use oncolytic virus surrounded by a cell membrane material, and we use it for immunotherapy applications, and we demonstrate that this construct works much better than the oncolytic virus itself. It induces a much stronger immunotherapy or immunoresponse as compared with the virus itself. Here we use a mesoporous silicon material, nanoparticle, that we fed to cancer cells, and they actually were wrapped with the microvesicles. And then when we inject back, we demonstrate that they could be guided to the cancer tumors as well. Recently, we have used microfluidic technology. I know that you are also working on this. So here it was not to construct or to get the best particle possible, but to get the highest amount of drug possible inside of, in this case, a microparticle. So we got a loading degree that was almost, it was 80% loading degree by using this kind of technology. And then recently we did a screening formulation, and I will focus a lot on this work that we have done. There is a nanoformulation for cardiac therapy, for cardiac targeting. I will come back to that later. But as I said, we have also used other materials. So for example, in this one here, we combine inorganic material with cell-based structures, and we use it with gold nanoparticles, and we use it for NIR therapy, chemotherapy. So it's a combination therapy. We have also used here, in this case, MOFs. So we synthesize MOFs, metal organic frameworks, and we also use it for microenvironment tumor regulation. And we also, this is one of that I hope I have time to show you, where we combine, well, this is a liposome or a lipid nanoparticle that we put cytochrome C inside together with a small drug molecule for cancer therapy. And this one here, for example, what we did, we extract the cell membrane of the blood, so exosome-based structures, and we load them with magnetic particles, and we did a magnetic guided therapy for cancer. I will come back to that later. We also are looking into new fabrication methodologies because the field is evolving a lot. So part of the group is also working with 3D printing structures and microneedle structures. So basically, we avoid these injections of nanoparticles because we know all the problems that are there. So what we do is that we also load this, for example, small microneedles like these ones, with either with EVs, extracellular vesicles, for example, or with other kind of materials, and then we just use them as a patch under the skin. So, oh, sorry. Oops. So we can also do that, or then with small patch. These are implantable patch, so you can put it, for example, on wounds, or you can put it on injury tissue that they can contain nanoparticles, but then they are local delivery, so they are not injectable, for example. So these are other possibilities. But yes, we have done a lot on microfluidic technology. So we have a few patents on this, and we have also our in-house microfluidic system. Is very different than the commercial one, I can tell you. I learned this technique when I did the part of my postdoc in Harvard Technology. Professor David Witz had a very big lab, and he still has a big lab. And basically what you need is just pumps and the knowledge how to do these microchips, because these microchips are very cheap. It's like less than 10 euros. You can do, and you can use it many combinations and use it for whatever you want, basically. And with this, we can do a lot of microsystems and nanosystems. And then we have done quite a lot throughout the past, including scaling up. So this is a paper where we demonstrated for the first time the scaling up. At that time, we were able to do about 200 grams of nanoparticles per day. And nowadays, we can go up to 1 kilogram per day by using this microfluidic technology, particularly of polymeric nanoparticles. That's what we have done quite a lot. So let me start with cancer. So as I mentioned, we can use this kind of constructs, microneedles, and we can load them. Well, in this case, it was with a virus, but it could be particles. We are also working with the extracellular vesicles that we load them and then give them on top of the skin. So we call topical application. And it works very nicely. I don't have time to show you, but we just published recently one of my students, and we show very nice how we can induce immunoresponse by doing this kind of strategy very, very strongly. This was the one that I was telling you before, so that we extract the EVs from the blood, from the mice, basically. We load these EVs with doxorubicin and supermagnetic nanoparticles, and then we inject back to the animal. And here you can see, basically, we guide them with a magnetic field. So here you see. and we were able to see the accumulation into the tumors and a very strong reduction on the tumor volume of this. So this was one of the, and the survival rate, so you can see here, a survival rate over the several days. So this is what we try to do with our formulations. Here we demonstrated, as I already mentioned before, so we use it for breast cancer, and basically it's a combination of, it's a cancer vaccine, it's a combination of chemotherapy with immunotherapy and NIR irradiation. And what we demonstrated, this is just an example, but what we demonstrate is that if we give this kind of vaccination before the animals have the cancer, when then we inoculate the cancer, we saw no cancer tumor development. And when we do the other way around, so if we give this technology or this kind of vaccination after the cancer, then we see a much worse result. So this tends to tell us that perhaps in the future, maybe in those families where the cancer is very common, maybe a pre-vaccination of individuals could be one of the solutions to prevent the future of cancer, for example. So this was a very, very nice study that we demonstrated as well. This is with the metal organic framework. So you see, so this metal organic frameworks or MOFs are very interesting. Why? Because when they are separate, they have a certain behavior, and when they cluster, they have a totally different behavior, which is very, very nice. And we realize that if they are separate, they are very stable at a physiological pH, but as they cluster inside of the cells, they became extremely toxic and they can kill the cancer cells. So we did this, let me go back. We did this by microfluidic technology. So we created these MOFs by using synthesis, using the microfluidic technology. You end up with this kind of construct. And if everything goes fine, you see, again, after we gave them to the animals, we saw a clear reduction in the tumor volume, but more importantly, we saw a regulation of the tumor microenvironment. So what does it mean? This means that your cancer gets more leaky and you can actually tackle the cancer much, much easier than the normal drug molecules, for example. So let me show a little bit of one of my particular interests, that is the heart. So we call it, we have several projects on this that we call how to restore a broken heart, basically. In this meaning is really how to get the heart a new life, because unlike the cancer, you don't want to destroy cells, you want to regenerate new cells. And that's extremely difficult when we are moving to the heart. It's really extremely, compared to cancer, of course, because we already know a lot of the cancer. And with the heart, we are baby steps learning how we can handle the disease. By the way, cardiovascular disease are still the #1 in the world. So most of the people still die because of cardiovascular disease when compared to cancer. So it's a very complex disease. There is an infraction. And the thing is that if you don't act, especially if it's an acute disease, if you have about few days to treat the patient, and if you don't, you lose it. So while the cancer is a progressive disease, it can take a long time. This is very, in acute cases, so in 1 week, you might, if you don't act, you might lose the patient. So what we've been trying to do is, again, you see here, so these are porous materials. You see the porous materials. We pegylated these particles. We modify with the targeting peptide here that we call the GPS. And these stars here that you see, they are radio-labeled materials. So we radio-label these particles. And the red are the drugs. And we injected these particles and we wanted to get it to the heart tissue. So this is the myocardial infarction, the models that we developed. And this was one of the very first studies. And we could see, this is imaging, this is a rat. And what we could see is accumulation of these particles into the rat, about 60% of the injected dose, which at that time was really nice. But as you can see, unfortunately, well, you always see a lot of particles accumulating into the into the liver and the spleen. So, well, we left these particles and start to think another ways how we could use it. So we have done quite a lot lately on materials. So these are what we call sugar-based. So they are actually beta-glucan particles that we have done it in the lab. And basically these particles I will show you, but basically we don't need to target directly there. They can attach to monocytes or macrophages, and then the macrophages will bring them there, because when there is inflammation, we have certain cells that go there, and then basically they release the particles, and then we can do a lot of different things. So this was actually my former PhD student that came up with that kind of idea. He designed these beta-glucan nanoparticles. He loaded them with different proteins. And what we were expecting to see is that this beta-glucan would attach to this receptor very precisely of the monocytes, and the monocytes will bring to the inflammation site, release the particles, and then, reduce the inflammation, basically. So you see here, this is a heart of the animal. We developed models to make sure that it was working properly, like in humans. And then if you see here, so this is the hard tissue. And if you see these blue lines and the thin tissue means that it's not good. And as we move to our formulation, you see these are the thick walls, then it means that our formulation has worked as it should. So we got the particles there and they are working as they should. And this just came out actually. So this was just coming out 2 days ago. So we started to think that maybe there is an influence of the spleen. So we know that a lot of particles end up in the spleen, but maybe we could use the spleen as a motor to get our particles elsewhere. So here, now is my postdoc, but this Zewa had a crazy idea. Why not if we remove the spleen and we try to understand how the particles actually can get to the heart and lead to a reducing inflammation. So in other words, what the monocytes or the macrophage that you have a lot in the spleen, how they can influence. So we did a lot of studies. I don't have time to show you so much, but basically, first of all, well, he removed the spleen from mice, first of all, and then we start to study it. And what we did, so we created what we call a construct that has spleen target. It's called a nanoerythrosome that is engineered with a targeting peptide. So this is the targeting peptide. Don't mind. We also I don't know if phase displays is a technique that you are familiar. So you can identify receptors in the human body or in animals by using bacterium. You inject the bacterium gets to certain receptors, you isolate those receptors, and then you construct peptides that are specific for those receptors. So we use very good collaboration in the University of Tartu. I was there 2 weeks ago. And basically this is one of the peptides that we found out for the heart tissue. And then we use a small drug molecule, but basically you can create these very beautiful nanoerythrosomes that you can then, well, you see here some of its properties that you can also then inject and see if they are going to the right places. And that's what we did. So we use, of course, a lot of fluorescence-based materials. So they are fluorescent labeled, but you can see here the accumulation. And at this time, we actually want them to accumulate into the spleen, because that was our main target at this point. And then when we look, even when they accumulate into the spleen, our surprise was that they still have an improvement in the myocardial inflammation. So they were still able to move towards the inflammation, or move from the spleen back to the tissue inflammation. And if you look, so these are very complex diagrams, but basically this is an echocardiogram. I don't know how many of you have done it before on yourselves, no? So this is how it looks like, but basically you want something that looks more like this. And again, when we do the histology, you look into the tissue, so you see the tissue here, it doesn't look good, and here it starts to look as normal as possible. And then when you look into certain areas, so for example, fibrosis, you see a decrease in the fibrosis. So this is what you actually want to achieve when it comes to the heart infarction. So we started to think that, okay, we can do that with small molecules. Let me see if we can do it also with, with gene therapy. So now you probably know that is very nicely, very well established. So we started with something simple. So using, again, our sugar-based structures, we loaded with sRNA, and we tried to see if that can again be used for inflammation. And we used 3 models of inflammation. So we used the heart, we used the liver, and we used the lung. So I will show you only one example, but basically it works for the liver very nicely, for the lung not so nice, and for the heart, these are the results that I will show you. So first of all, no toxicity in any of the other organs. So even when they accumulate in the heart, they look very nicely. But more importantly, so if we have here the inflammation in the heart, after we give our injections and then collect the organs, so you see the healthy model, they all goes to the liver basically. And in our model, you see that still in the liver, but also in the heart. But most importantly is here, so it's the accumulation in the inflammation heart is much higher. So how does it translate in terms of inflammation and gene silencing, basically? So we want to observe is this protein expression is going down, and the collagen production is going down. This is for inflammation what you want to achieve, basically. So when we see also into the tissue, you see the tissue behaving more like a normal tissue. So that tells us that our therapy is working, even if some of the materials are not getting there, so they go to the liver and spleen. It doesn't matter, because those that are getting to the inflammation, they are getting a response that is sufficient to induce the reduction of the inflammation. Obviously, then we didn't want only to stick with one material, so these beta-glucans. So we started to think, can we find other formulations or combination of formulations? And that's where it started to get much more complicated. So, and for that, we design what we call a high-throughput microfluidic platform formulation. And that is basically an automated formulation where you do microfluidics, you do robotics, and you do particle characterization all in line. Okay? So basically, then you can build, well, we use it here as an example as Legos, but basically you can do combinations of different materials and then end up with the material that is the best formulation possible that you want. So here is what we did. So you can see how it is our microfluidic design here. Then we have a microplate here, so it's automated, and immediately the robot analyzes each one of these formulations by DLS. So this is done in one single go, basically. And then you can try different polymers combinations, you can try different small molecules, you can try different targeting peptides, and you can try different lipids. Some of these lipids we got it also from a collaborator. You probably know Dan Pear, so he gave us some of these lipids as well, so that we try, because we already saw in the literature that he has published a lot with this and they were working, so we thought, why not? So then, well, you see here, so just to show you some examples, some of these are the formulations that the robot checks. And here you see all the combinations that we did in terms of size, in terms of the zeta potential. And then if we have a blue dot, then it means that it's a good formulation. If we have a gray dot, then it means it's not a good formulation. So we don't even continue with that. So by doing all of this, then you end up with still a large kind of formulation. So then we need to discriminate. And that's what we also use a very nice computational analysis, because then with all these parameters that we have here and this computational analysis process, we can look into the different parameters and then get to the conclusion which ones we actually go to the in vivo. Okay. But of course, to get this, we have to do all that work that the microfluidic robot automation does. So just to show you some examples, then we got one particle. So here is the particle that we got. And this is the particle that we went to test first in vitro to make sure that it is working. So here I show you some of Western blots that shows that the signal was active and it could be work. And then after that, we go again to in vivo. So again, you can see more accumulation here into the hard tissue. And when we compare with the healthy tissue, less. And when we have the inflammation, much more accumulation. So this is how we design our nanosystems. And again, you see similar plots as before. So we look into the inflammation over the time. So 7 days, 28 days. And basically, as we progress in time, you'll get a much normal echocardiogram system. And when we look into the histology, again, you get into tissue that looks more like the normal tissue of a heart, of a healthy tissue, basically. So this is how you can use materials, but you also can take materials that are not so well yet characterized, like MOFs. Is anyone working with MOFs, by the way, in this group? No, with porous materials. So they are very exciting materials, by the way, because you can do just by chemical coordination, you can do beautiful kind of structures, very well organized, very well arranged. You can do cubes, you can do cylinders, you can do whatever. And they are porous as well. So you can load also drug molecules inside of these porous structures. So we did very simple is we synthesize iron-based MOFs. So they have this kind of coordination structures, and basically they have these pockets where you can put there whatever you wish. And you see here the size is very small as well, the charge of this, and they have a very well-defined X-ray diagram, so you really can identify if they are or not MOFs. You see here the structures of this. These are like hexagonals, so you can see how they look, and you have this beautiful and always very well similar structures when you do this chemistry. So it's very repetitive, so it's very easy to get this. And then, of course, you can also attach whatever you want on the surface of this. So we wanted to actually, what we wanted to do is that, let me go back, sorry. So we wanted to check, here. We wanted to check if these materials, if we load them also with RNA, for example, first, if the RNA still is active, and then second, if these materials can still deliver the RNA inside of the cells of interest. So that's what we did. So we loaded sRNA, and from these pictures here, you can see that basically it is working very nicely. So it is stable, it is not degradable. We get a very high encapsulation efficiency. So you can see here, almost 100% of this. And here you can, in vitro, we got a very nice gene silencing efficacy. So this was one of the things that we can demonstrate. And then in this case, we use it for arteriosclerosis. So basically, sorry. So basically, what we so this is not anymore at the heart tissue, but is in the veins, let's say. So you can see here when there is a heart failure is usually because the veins they get clotted with fat or with other kind, they got obstructed. So basically, if we can reduce this, then obviously you can prevent the heart to fail. So what we demonstrate is, again, we create this model, and what we can demonstrate is that by using this kind of a strategy MOFs, they can accumulate into these areas, and by releasing the sirna, they can actually decrease that blocking of the veins, and definitely prevent the cardio failure, basically. So all these models, they take time. So as you can see, it's a lot of manufacturing characterization of the particles, in vitro and in vivo assessment. So it takes a lot of time. So my group has also been working a lot on models that we could assess immediately the fate of these materials. So here I show you just one model where we did, this is for cardio, but it could be for cancer, it could be for other kind of tissue, it doesn't matter, because we use iPSCs, so cells that we derive from patients, and then you can derive it in different kind of organs. So Raquel, my PhD student, what she's been doing is doing this kind of, this is basically, it's cell-derived, it's a cardio tissue deriving from cells, and they behave exactly like the cardio. So they beat, they have all the composition as in the tissue, exactly the same. But then you can do it in a very small scale. You can give particles to this, and then you can also create a model so that behaves like in the patients, basically. And what you can see, or I don't know if you could see, probably not. Let me see. Let me go back. They should beat. So this is how it should beat, like this. And this is when there is a problem with the tissue, it beats very slowly. At some point it will beat, but basically this is what happens with the real tissue. So when we give nanoparticles, and in this case, let me go to the next, here, so we treated with nanoparticles loaded with microRNA in this case. What we see is the accumulation of the particles in the tissue. So here now we are looking at the cellular level. So we don't need to do the in vivo, we just look into the tissues and we can already understand, because those are human-derived tissue cells, and then we can already understand in the tissue how these materials, how these formulations work. Well, these are just some conclusions from the heart. So we have a lot of projects now that are going on in the heart tissue because we really want to understand more about this technology, but it's not only about that. So we have been also working on large molecules. So now when we move to proteins, it's a totally different story because they are very large molecules. And we have faced a lot of challenge to get these proteins actually inside of nanocarriers. So my student here, so she just defended in November the PhD thesis, shows she was working with different kind of materials, lipids and polymers, in order to encapsulate proteins, or in this case, it was cytochrome C, inside of these constructs for cancer therapy. It turns out that it was not so easy. It took us a lot of time to develop this formulation, but at some point she managed. But you can see that there are a lot of proteins that you can consider that you could use it for different therapies. The problem is that they are very challenging to formulate. So we did a microfluidic system where we use LNPs, as you can see here. And after a long time, we were able to encapsulate cytochrome C together with this small drug. And what this combination does is that when they go inside of the cells, they induce apoptosis. So basically, we were able to study all the mechanism from the intracellular, from the binding till the particle goes inside, and also all the cascade that happens inside of the cells, up to the cell death, basically. So this is what we did. I'm not going to show you all the process here, the in vitro and the in vivo, but I just want to show you basically, let me go here for the in vivo, maybe it's very relevant, but so just to show you, we did different injections, and you can see here the tumor growth goes down, it is decreased the tumor volume. So demonstrated that basically our formulation, and in this case we target again with a peptide, so we modify the surface with this peptide, and we can modulate the ratio of this modification on the surface, basically, but with anti-cancer or a receptor binding peptide that we identify for cancer therapy applications. So just to show you that it is possible to also achieve protein delivery, even though it's a little bit more challenge and the particles fabrication. So sometimes you don't have to invent the wheel. So if formulation for injection becomes too complex, then maybe we should think about something else. So what we have been using, or some of the strategies that we do sometimes, is that we do local therapies. So we basically created, or in this case, we create Prussian blue based nanoparticles with manganese oxide. And these particles we actually incorporate into a hydrogel, and then we inject this hydrogel directly into the tumor. Of course, in this case, they are topical tumors, but basically in this way, we don't need to carry so much about the biodistribution of these particles and so on, because they are on the surface. But the idea here is, again, to kill the cancer tumors, and again, it's based on the properties of these particles. So they are, of course, reacting to the laser. Usually we use 880 nanometers laser. And with this fabrication, we also demonstrated that, again, you could, after you add it with the light, with the laser, with the NIR laser, you could reduce the tumor volume, you could increase the temperature, the local temperature under the skin, and that has an effect because that allows us to release the drugs. Some of the tumors even disappear, and we could monitor the glucose concentration levels that in some cases has importance, because in this case we were using glucose oxidase, and the glucose oxidase we actually were helping in order to bring down or to intracellular effect in order to decrease the tumor size as well. So these are strategies that you can also use. So we are becoming more and more fans of local injections. So again, this is just to show you what we did. We call glucolysis modulated hydrogel. So again, we prepare these microspheres that we loaded with metal organic frameworks, loaded again with big proteins, so glucose oxidase, and then we inject this into the tumor, and then we follow again all the cascade into the tumor by using again NIR laser irradiation, and then we can demonstrate. Well, first of all, let me show you. We produce these beautiful materials here. We put them inside of these microspheres. These microspheres are degradable microspheres over the time, and then and you can see also here the wavelength of this, so the absorption of this, intensity, and so on, and they are reacting to different pHs. And then when we look again to the animal tests, so again, after injection, local injection, you see a very much decrease of the tumors. And this is, again, a way out to do it if you don't want to do an injection of the nanoparticles directly into the bloodstream. So for surface cancers, this can also work very, very nicely. And even in this model, actually, we also tested with a metastatic effect. And when we could see that even in injection one side, after we inject our particles, were able to also tackle other cancers that were growing elsewhere in the body of the animal, and they were able to, as you can see here, to reduce the metastasis of the organ of the animal. I wanted to finish here because I think I show you quite a lot already of what we can do, and then I wanted to also hear some of the discussion as well. Of course, you see here a lot of different kind of project that has helped us to do a lot of this work that you see here. Is a lot of people that is involved. Is a lot of financial support. Of course, we are working together on Marie Curie Rise as well in this, but there are many others that we have trying to collaborate, and I'm looking for to other collaborations in the future as well. And of course, let me just show you. So this is the group that has worked a lot, and I didn't even show half of that in Helsinki. So this was my lab in Helsinki, and this is my lab in Netherlands. Most of the work that I show is of Gao. So she just defended in the beginning of January the PhD thesis on heart. And these are some of the key collaborators on that we have all over the world. So Bruno from Portugal, Vince from Finland, the cardiologist, so a clinician cardiologist. Tembat is from Tartu, so he does all the targeting peptides for us. And then some of our collaborators in China, in Shanghai. So they are also helping with all the animal testing as well when we need, and from Mexico, Yareli, from other kind of projects as well. So this is the group. We have about 22 PhD students. They probably are not all here, and several postdocs as well. Monica was a visiting PhD student from Zaragoza. Idaire is my postdoc, Marie Curie postdoc from Tenerife. So we have a very international. Mainly, we have, I think, maybe 8 or 9 nationalities or 10 within the group. And we are also very much looking for visits from different backgrounds and PhDs. But also, if after your PhD you have nothing else to do, or you want to experience something else, you are very welcome to apply for Marie Curie postdoc fellowships. Today, this morning, when I opened my email, I got the news that we got 3 this year, so 3 new Marie Curie fellowships. So the Marie Curie fellowships are for mobility, so you have to move from one country to another, but it doesn't mean that we cannot continue to collaborate even at that time. And for example, with the Idaire was like this. I have here also other. At the moment, I have 3 in my lab. So, but that's how we can also enhance the collaborations, and also gives you a nice 2 years of safe that you can think what to do next, and also build up your CV as well. And then maybe later you can come back or you can stay. It's up to you. Good, and I'm then happy for questions. Thank you very much for your attention. I try to keep the time.
Yes. Is it working? Yes. Okay. Thank you, Hélder, for this very nice and impressive overview of all the work, all the materials developed. So now it's time for questions, and we have some questions in the audience.
Thank you very much for a very impressive presentation. I just want you to comment. You mentioned at the beginning you started with inorganic, like a porous material, and you shift to polymer. So I want you to emphasize what's the big difference when you do with two these. Some are working good or in general? Thank you.
Thank you. That's a very good question, of course. So when we start to use the porous materials for biomedical applications, it was in terms of stability and in terms of reproducibility was complicated. So we got a lot of these materials because they are also a little bit heavier. So in terms, they are more dense. They don't tend to perform as well as we expected when we inject them into the animal models, for example. So we start to have a bit of difficulties for the translation of these materials, and that's why we end up going for polymer-based structures or lipid-based structures, because at least for the translation, they seem to be better, stable, and also faster to translate. It doesn't mean that the others will not work, but it requires a much longer time and effort to make them more suitable for biomedical applications. That is the reason that we put a little bit aside, and now we are trying to move from those materials to these metallic organic frameworks, because at least we get a better control on their structure. Because one of the problems with the silica silica is fine, but with the silicon is their morphology and structure is very random, for example. And we don't, or at least the ones that we fabricated they are not as beautiful as the one that I showed you, with very well-defined structure, very well-defined properties. So that was one of the reasons that we left them a bit aside and start to work more on the other ones. Yeah, but they can be used for other kind of applications.
Yes. Just one thing, because I was also wondering about this and following up, because you also did a lot of mechanistic studies.
Yes.
So with this, I was also wondering, for example, with the lipid nanoparticles, when you do delivery, you can, let's say, trigger the release from endosomes also due to fusion with the lipid endosomal membrane. So I was wondering, when you compare with, like, for example, the MOFs, or have you studied this? Like, how is it in that case that you really achieve the delivery to the cytosol and the release from the endosomes? Have you looked into that?
Yes, so it really depends on the properties of these materials. So with some of the materials, so for example, when I showed these metal organic frameworks, what we observe is that they were very fast going to the cytosol. But that's because the material itself has properties that ruptures when they cluster, they kind of blow up the endosomes very, very fast. So that was very, very nice. With the others, you can do that, but then you might need some extra help, or then you need to use other kind of pH regulators that allows you to get out of the endosomes or lysosomes. So it really depends on the properties of your particle. And in some cases, with inorganic materials, usually we find it much easier. With lipid and polymeric, you have to use a mixture of, for example, pH modulators, so that you can get out of those. With inorganic materials, with the chemistry, we can achieve that in a little bit better way, or faster way, let's say. You can also do it with the other, but with this, it's faster.
Mm-hmm. Okay, thank you. Interesting.
Welcome.
Thank you, Hélder. Very nice talk, overview, lots of nice results.
Thank you.
So if I properly understood, the materials that you produce mainly are produced by co-injection or by microfluidics?
Mainly for microfluidics,
Okay.
yes.
So my question is, because also you did a lot of in vivo testing.
Yes.
So when you go to in vivo testing, do you continue to produce the materials also by microfluidics, or
Yes.
you change?
Yes, yes. So we do all the fabrication with microfluidics, we do the characterization, and we use these materials for the in vivo application.
With the idea to arrive to first in human also with microfluidics?
Correct. So the idea is that you could then scale up this, because, well, microfluidic. So we are working on batches that they are, let's say, if we talk about in mass, few milligrams, 100 milligrams that we can produce. But the idea is that you could scale up then to a larger amounts of particles. So the idea is that you use that setup, exactly setup, for a larger volumes if you would like to continue to a clinical trials, for example.
Okay, very interesting.
Yeah.
And also another, no, also in the this system for screening, I found very, very interesting.
Yeah.
So it's a curiosity, for example, more or less, which is the number of different formulations that you can produce?
Oh, well,
for
a lot.
On one day, and also because if I properly understood, you use the LS for the first go, no go, no? If the particles are produced or not?
They are stable, if they are big, and so on. Oh, a lot. You can do a lot of formulations per day, yes. Because you have different combinations, and it's on a 96-well plate. So you can do a lot of different combinations already there. So per day, you can do a lot of formulations. I don't know the number now, but it's a lot. That's beautiful of that, yes.
Thank you.
The limit is that the number of components that you want to put in those formulations, because if you want to to put, I think it was more than 5 or 6, then it's much less. But if you want just 3 or 4, then it's you can get a lot of different combinations in one day. Thank you.
Hello, thanks for your presentation. It was very interesting.
Thank you.
So I was wondering about the MOFs, the metal organic frameworks. Well, I don't really know much of how they work themselves, but if I understand it correctly, it's like you can synthesize them so they can be very specific to the molecule of your choice. So they have like a binding domain that's very specific. Yeah. But then
morphology and the morphology as well. So you can make cubes, you can make hexagonals, you can make triangles. So it's very, and it's in terms of reproducibility, they all look very beautiful the same.
Yeah, so then I was wondering, like, how do you exactly use them? Because for what I understood is like you create these MOFs, and then you load the drug of your interest inside the MOFs.
Correct.
But then the MOF that's himself, you load it inside the nanoparticle.
No,
or you use
not
it
always.
as the
No.
nanoparticle.
So you in this case, what we did two things. In one case, we loaded inside of a microparticle because we wanted the local injection, and in the other case, we just used them like that.
So they can be used
as nanoparticles. They can be,
yes.
If they are stable, and if the material that you are loading is also stable, then you can just use it like that. Yes.
And they will unbind from the protein that's loaded inside, as the same as like a nanoparticle.
Yeah, it's exactly the same. It works exactly the same, but of course, because again, these are inorganic particles, they will also tend to go a lot for the liver and spleen. So they are less. That's exactly they are less stable than they are, for example, the LNPs or the polymeric nanoparticles. So that's the only problem. But in terms of shape, in terms of structure, they are much more exciting because with a simple synthesis, you can get them very widely reproducible, and also you can scale it up because you can also do this synthesis with microfluidic technology.
Okay.
So there is pros and cons with like in all kind of materials, and it depends what you want to do. But if you just want to use it as a delivery system, that is also possible. Because the pores are really very small and very well defined. These pockets that I show, yeah.
Thank you.
Thank you.
Thank you for the presentation. Very nice. I was wondering about the cancer applications, the different examples that you gave. One of them, it's the experiment that you take out the spleen and you still...
That was for heart. That was for heart.
Okay, for the heart. But
yes.
Okay, then related to the immune response, did you study the response from the bone marrow? Would you expect that the immune...
No, we did not know. No, so our hypothesis was that whatever is in the spleen, it can also help, well, like the different kind of macrophage, monocytes, the immune cells, they could help to actually get an effect in the heart. So even though if the particles end up there, they can still perform. So that was our hypothesis. That's why we removed the spleen to actually evaluate that. But we did a lot of the immunoprofiling as well and study how that it affects the response. So we did all the genetic profile as well, but we didn't study the bone marrow or nothing like that. No, not in this case.
Yeah, and similarly to the cancer one, where you show the vaccine effect, right, that you were in.
Oh yeah, that we did.
Okay, I was wondering if in that case, you would see a similar response in a bilateral tumor model, like to mimic metastases, for example. Would you expect that also,
That's a very good question.
Treating one, you would see an immune response to another one? Or?
That's a very good question. Well, I cannot say how it would work because in that case, we only studied one single cancer. So that was melanoma. So we don't know if you have metastasis, how it affects. And we also don't know if you have a breast cancer, for example, if that would differ, because we only use it for topical cancer applications. And there we very, well, I didn't show it in here, but we did all the immunoprofiling. So we have, if you go to the paper, we have all the dendritic cell analysis. We have even the T cell production analysis and so on. So we were able to identify which cells are actually more proliferating and helping to tackle the cancer. So, but we don't know if a different cancer model will lead to a different result. We don't know. This is a technology that we also initiate as a startup, and now we are trying to develop. But we believe that it's not so much dependent on the cancer that is used to, but is more how you modulate these structures that you have inside. In this case, it was the microneedles that you are referring to, right? Yeah. So that really depends on the materials that you have there, because they can actually, the combinations that you use, they can already help for immunomodulation. Yeah. Yeah. Exactly. Yeah. Thank you.
Okay. So maybe Anna.
Thanks for the widespread of techniques and materials that you have shown us.
Thank you.
And I wonder if any of those, do you think that they have shown promise to be translated to the clinics, and which one, and how you expect the regulation for that particular one?
Very good question. This is always the questions like which child you like the most, right? Let's be honest. If we want to do the clinic translation, we always look into materials that are, in a way, in the process that you have a clearance already from the regulatory. The regulatory knows already very well these kind of materials. So now we are putting a lot of emphasis in LNPs. So in lipid nanoparticles, because those are the ones that, well, a lot is already out there. There is FDA approved for many of those. So we think that those are the ones that will be faster to be translated. Now, if you want, and you can use it with different kind of modifications now. So, but if you want what is more exciting, I would like to see more of these inorganic materials to move forward and to get it to a clinical translation. But also when we discuss with our clinicians, they are always also very scared. So when you see inorganic materials, when you see all these particles like moths and silica, for them sounds a little bit too much to put in a patient. So they obviously think more on materials that are more bio-friendly or easier to use, like lipids, like polymers. So or cell-based structures. So exosomes, for example, is another thing that we are working quite a lot. Even though, again, exosomes, in order to reach to the next level of regulatory, it's also a mess. It's a nightmare. So yeah, it's never easy. But I would say that if we go with the LNPs kind of structures, liposome-based structures, those have the great potential to go faster to the market. Welcome.
Maybe also a related question to that. I was wondering, so do you think that also all the computational capacities right now will help to in the translation to the clinics, because there's like a lot of data out there, but still, like in general, they are very complex systems. Biological systems are very complex. So to have all those data sorted out, do you think then in the next 10, 20 years, this will help to get more nanomedicines into the clinics?
I hope so. That's a very good question, a very good point. And I hope it's less than 10 years, because I think, especially with those kind of materials or these kind of particles, there is already a lot of information out there. And well, you probably, as you know, machine learning can also help to collect a lot of this information in order to construct. So the more we have studies and information about those, the faster it will become in the future, that's for sure. And that's one of the downsides of the new materials, is that we don't have enough information to make a drastic conclusion that, yes, this is going to be very effective. While with the other ones, that the more in vitro, in vivo clinical studies we have, the faster we can make the next generation and the next generation. So, but of course, that's why I say that I still like the inorganic materials, some of the organic materials as well, because if we never collect that data, we never get to that point. So someone has to do that job, even though I know many groups that they don't want to do that because they want to move faster anyway to the clinical setting. But if we don't do it, then we might be wasting some of these materials that are really interesting and have electronic properties. They have whatever surface properties that they are really, really, really nice. But just because we don't have enough studies, we cannot consider them to the future. So the more people are also joining forces, the better it is for getting that data. Thank you.
Okay, well, thank you, Hélder,
Thank you very much
for your talk and answering all the questions. So thank you, everybody, for coming.
Thank you.